All right, good morning, everybody. Welcome. What did I just do here? Exit, maybe. Okay. I got too many buttons up here. So we've got just a couple of slides left to wrap up with the eukaryotic organisms, a little bit about wrapping up a little bit about some fungi. We'll talk briefly about some lichen. And then we're going to shift over into the to chapter 6, which I want to say is viruses. Most of it is, but really it's acellular pathogens, because there's some other stuff that aren't virus that we will think about as well. So we left off talking about a handful of different types of fungi, and really we're just looking at some of the mechanisms as far as how they are able to reproduce. So some, this always I mean, it's microscopic, but it looks like kind of peas in a pod. These ascospores are the peas, if you will, and the pod is this ascus, and it is the reproductive mechanisms for some fungi. They in turn open up the ascus, releasing the ascospores, and we get more fungi, more hyphae continuing to grow. So here we could see just a cartoon with this, and we can see, like we talked about before with the fungi, when we talk about sexual replication with these organisms, it's not male and female when we're talking like that, it's just opposite mating types, right? So slightly different variation within the two strands of hyphae that in turn allows for the fusion of the two. And that's what we're seeing right here with this. And you get multinucleated, it's dikaryon that is diploid, if you will, all right? And then creating the haploid ascospores within there that get released and the cycle continues. Basidiomycetes, Basidiomycota, these are the mushrooms, all right? This type of fungi generates a plethora of different types of fruiting bodies, these basidiocarps, that have the spores within them. Similar to what we've looked at, these haploid spores get released, settling down into the environment, growing as a mycelial mass or the hyphal threads, breaking down nutrients within the environment and pulling those things in. There are all sorts of cool things when we think about some of these. And in biology, we talk a little bit about, you know, just diversity of organisms. And when it comes to thinking about like some of the biggest things on the planet, you probably think something like a whale or something like that might pop into your mind. But deep down, it's fungi. There are in the, I think it's Oregon, maybe it's Washington, some of the forest areas in there, they recognize this type of fungus that is massive, like 4 square miles or something like that, all under the soil, and it's all ultimately one genetically identical fungus that has spread over the course of a, I don't know how long, super long time, all right, that's lent to its potential. You also can see some stuff around here in the spring where we see mushrooms popping up or toadstools whatever, some of these fungi. And maybe you can see them like in your lawn with maybe kind of an area like a ring. We call them fairy rings. And if you happen to see some of this stuff and you pay a little bit of attention from one year to the next, that ring progressively gets a little bit larger. The fruiting bodies, the mushrooms, show up at the periphery. Underneath, in the center of that circle, is this huge mycelial mass that's all underneath, all right? And it continues to progress. So you could kind of envision something like that in the forest floor over the course of thousands of years, having the opportunity to spread and become one massive organism. And to me, that's pretty mind-boggling. There are all sorts of potentials with these guys, many of which we think of as being possibly edible and some severely poisonous. And some of that, they look very similar. So I recommend if you're going to imbibe in this stuff, you probably ought to just be buying them from the supermarket. Otherwise, it's a chance that you don't know what you're truly coming in contact with. And this Amanita is definitely a potential for some serious issues. So here, this just shows a handful of different potentials, and some of them that we may think of as potential problems, or maybe something that we might tap into as something being edible. So we can see here some edible mushrooms, morels, truffles, moving down further, others, more mushrooms, and definitely issues. Cryptococcus neoformans is one that we fit within this. That is, although we're talking the Bacteriophages and these fruiting structures, crypto is more, as we look at it microscopically, yeast, definitely different. So this showing some more details with this trichophyton, we will get into later. This is one that we see as like ringworm situations. These dermatophytes cause all sorts of potentials for fungal infections in the skin. And there are numerous types of dermatophytes, things that target the skin. And so there are kind of handfuls of things that we could say are the culprits. This is one of them, but there are more for fungal infections in the skin. And we'll touch on some of that a little bit later. All right, shifting over, here's a dinoflagellate, which has some odd morphologies, a cellulose sort of armor or shell, and a couple of flagella between the cellulose plates. So kind of an interesting one. The next, we've got all sorts of different algae and diatoms, all sorts, some massive and some microscopic. We talked a little bit about like the Winogretsky columns and potential for stuff like algae and others potentially maybe blooming in there if it has lots of sunlight. You also would be pretty likely to find things like these diatoms. And in organic gardening, the dead shells of these guys are used in organic gardening as diatomaceous earth. It's like they're almost like little shards of glass that gets into the insect and destroys them. Chlamydia modus is a unicellular algae. You can see some chloroplasts and flagella within them as well. And lastly, the lichens. You have all probably seen some of these guys. I think we've got an image here in the next. Some of this lichen. And it is a symbiotic relationship where we've got numerous organisms participating together, all right, as they all kind of form this community, where we can see numerous participants with some photosynthesis up here. some fungal and bacterial players as well often fit into these symbiotic relationships. And you get a handful of different types as we look at them. And this, often, you see this on pine trees, and I think moss, but it's really not. It's a type of lichen. Mosses are different. And this definitely has some toxins. It says they've made some poison arrows with this. There are other compounds that fit that as well. All right. Enough of those guys. Oh, I probably was supposed to click and share instead of just closing it out. That's fine. Didn't like that. It's like, let's stop sharing like that. Okay, new share, new chapter. Maybe. Okay. So like I was saying, as we started this, I often, as we get into this chapter, really think virus. The big focus is virus, but there are some other things. So it's really acellular pathogens, things that we think all sorts of real potentials. There is a lot of talk about virus. Is it alive? Is it not? There are parts as we move through and think about defining life that fit. And then there are parts that don't fit. And that sets the stage for a little bit of argument. How, what do we do, right? The fact that they contain some nucleic acids lends to some thinking that's enough to call them living. But the majority say they're such bare bones, if you will, they aren't able to do anything else unless they have hijacked some host cell and manipulated it to do that virus's bidding in the first place. Makes them say they're really not, right? They're not alive. And then we'll talk about stuff like the prions. Those suckers are just protein. So that's completely different as well, right? So there are all sorts of different examples that we might utilize as we think about virus. We could throw our coronavirus up there and think about pandemic issues right now, but I don't want to talk about that stuff. I technically don't really want to talk about Ebola either, although I find it pretty fascinating. There's a video game called Plague Inc. There is all sorts of different types of things that you could be to in turn, you are trying to be one of these pathogens, and it's your job to really destroy humanity. Kind of cool stuff. And then also there's some of it that's a little bit disturbing, right? If you've ever played it, you know you are doing good if all of a sudden the Olympics have been canceled. You're like, sweet, something's happening. And with not the Winter Olympics that we're in right now, but the Summer Olympics, as they got canceled because of the pandemic, I was just like, holy cow, this is like Plague Inc. We're in some serious bad shape. If they're canceling the stinking Olympics, buckle up, friends. The end is nigh, right? That's what I'm thinking. And I'm like, holy cow, I'm putting this game away, right? We've got the board game Pandemic. We played that as well. We're like, we're in it, right? That's a different story. So I was playing this a handful of years back, and Ebola outbreak occurs. And it kind of just made me feel a little bit disturbed. And I was like, you know what? I don't really want to play this game right now. I don't want to be Ebola trying to kill humanity. That seems too much, right? And in turn, it's kind of just been mothballed now because I'm like, it's too real, right? I'm not doing it. But this is definitely something that comes to the top of my mind when I think about something like that, all right? And I also feel like we are super fortunate that the pandemic we are dealing with is not this epidemic, not pandemic. We would be in some serious, serious sad shape if this was the case. All right. So there are all sorts of details. You have all probably heard stories about Ebola, and some of it probably pretty accurate. Other bits maybe a little bit embellished, and that's maybe okay. All right. I feel like if it's something that makes you remember the severity, maybe that's okay. All right. So there are all sorts of potentials with this. And as we think about some of these crazy virus, the possibility for weird, novel, never detected things to pop up and plague humanity becomes more important as we think about our, surging population size and encroaching into habitats that were untapped. The possibility of coming in contact with things that truly infect other things, but now all of a sudden open up avenues to plague humanity is a real problem. And Ebola fits that scenario. So infection with this, highly contagious. Individuals find themselves severely incapacitated, bleeding, they say from every orifice, from your eyes to your toes. We could think about the potential for severe damage, but really eyes and ears and mucous membranes, the virus within the blood, just coming in contact with that is almost a death sentence for sure. So we see all sorts of issues with this, and this, a handful of years back. Ebola is something that still terrifies all sorts of countries in Africa, although we have seen a little of that in the United States, but nothing that truly became a huge issue, thankfully. The first virus that really was evaluated was the tobacco mosaic virus. And we can see an electron micrograph with some of these, and then some tobacco leaves over here that have been infected with the virus. All right. And so it causes big issues, but it also was, as I said, one that was the first that was able to be recognized. And there were, really research thinking about life in the first place, and DNA and proteins and all of these things. Viruses were evaluated with some of this as we were trying to understand all of these different things and these pathogens that were causing problems. What are they? Who knows? You can't see them, right? And this stuff, virus, was, and as you can see here, very, very small. It was able to be filtered through something that would basically trap other things. Bacteria and everything else gets trapped, but the virus does not. A filterable virus, something smaller, right? Some sort of crazy unseen pathogen, and still able to infect, right? And much, much later, we actually get the visualization. These guys are some of my favorites as far as virus go. I think they just look awesome. This is an electron micrograph. These are bacteriophage. So these are virus that target bacteria and specific types of bacteria, all right? You don't get a bacteriophage that just will infect any sort of bacteria, all right? There is serious specificity. There are definitely a little bit of, overlap. It's not just going to target like E. coli only, although some will. there are potentials that they may have a little bit of wiggle room and get a couple of different pathogens or bacteria that they infect, all right? And when we think about virus, lots of the virus really would probably fit within this category of bacteriophages. Bacteria are everywhere. And in turn, bacteriophage should also be everywhere, all right? You have them in your body. You have bacteria in your body. It makes sense that there would be bacteriophage as well, things targeting these bacteria, all right? They don't cause ill effects in you, all right? For the most part, all right? There are definitely some potentials with these as they may package stuff up, and some of it might be more than just their own genetic information, and in turn, inoculating that into a different bacterium might lend to some other issues, but that's kind of a special circumstance. We're not going to worry about that right now, all right? So when I look at this in here, this is a bacterial cell, maybe E. coli, all right? The cell wall right here, and then the insides of the bacterium. The bacteriophage lands on the surface of the bacteria, and it makes me think of some crazy sort of spaceship, some alien ship landing, and as it lands, it in turn, well, in this case, is going to infect. The probing happens, right? As the virus makes contact with that host cell, there are specific things that it is able to recognize with these tail fibers, basically sensing the bacterium, recognizing specific proteins on the surface of the bacterial cell. Part of the virus detects that and can detect if it's proper, right? We can infect this or not. And if it's not the right bacterium, that virus doesn't land. It's not going to dock on this bacterium. It's going to keep on moving, all right, if you will, all right, looking for the proper place, all right? And there's all sorts of cool stuff, potentially, with these phage and what they might infect, all right, and how we might be able to detect. Really pretty impressive, all right? So as this lands, the tail fibers making contact with the proper host cell, there is the sheath. This is kind of like a hypodermic needle. There are these tail pins, these little needles, if you will. And if we dock, and this is our right cell, we dock and these tail fibers make contact, the sheath is going to basically make contact with the cell, the tail pins puncturing in, and it's going to inject the viral genome. We'll talk about some details with this in a second. It's going to inject it through the sheath into the bacterium, and at this point, depends on the type of virus, all right? It's going to get in and start making more virus, all right? There are potentials for these virus to get in and incorporate into the bacterial chromosome and kind of go dormant, if you will. Every time that cell replicates, it replicates the viral DNA, keeps it in the chromosome, and now we get boatloads of bacteria that have replicated, and then we turn that gene on and we say, what we ought to do is make some virus now. And now instead of one cell making a dozen virus, now we've got hundreds of thousands of cells that all have this copy to make virus, and they get turned on, and now those hundreds of thousands of cells are making a dozen virus in each of them, blowing them up and releasing out thousands of virus instead of a dozen, right? Pretty brilliant strategy, all right, in order for that virus not only to persist, but to proliferate, all right? There are others that just get in and the bacteria just starts making it, fills up like a dozen or whatever, and the cell explodes, releasing those virus. So different ways those virus may potentially do things, but the end goal is the same. We want to make more of ourselves, right? They use the cell's machinery to build the protein to replicate the nucleic acid. It packages it all up, and then it gets released out, right? And these guys right here would go on to infect more bacteria, all right? So really pretty complex shapes as we look at these, very different, but same sort of an idea as, you know, looking at any of these other types of virus, all right? It's just depends on what they're going to infect as we think about that. So we've seen this before to put some stuff into perspective. And we can see our light microscope stuff and then moving over to the electron microscope to see things like polio or smallpox or the flu virus. We can't see these with the naked eye, all right? It relies on electron microscopy to give us images that we can actually see, all right? If we were looking at it with a light microscope, you would see our bacteria and you would see the whole thing and you'd see lots of them, not just a little fragment of the bacterial cell, right? We're zoomed way in. This bar is 50 nanometers, all right? So zoomed way in order to visualize these phage, all right? So all sorts of possibilities. And as we look at it, even though, you know, it always gets me here, the light microscope does not go clear here. I already mentioned this before. It's the same figure. Well, maybe not the same figure because I don't think we showed these on the previous one. So usually right around here is our cutoff with the light microscope, but about a micron. You can't really get much better resolution after that. So shifting from the bacteriophage, we're going to come back and talk more bacteriophage, but we definitely want to think about stuff that's more of an issue to a plague of humanity, all right? That's kind of what I think about with virus, but there are all sorts of possibilities with these guys, all right? So human pathogens, these viruses that want to get into us, it's a similar idea like that bacteriophage, where it's got specific substances, proteins that it recognizes, and then it's able to dock and infect that cell. When we talk about human virus, they don't just target whatever. I bump into a human cell and I'm going to infect it, right? It's got to be the right type of cell. Mucous membranes are great entry points to numerous types of viral organisms, viral pathogens. It's not an organism. That means it's alive, right? To these virus, they have the potential to gain entry, to dock to these cells. Others require other entry points or mechanisms to get in, all right? There are also differences when we think about the virus and how it gets out, right? How it leaves a cell, right? Some viruses, when they leave a host cell, will take part of that plasma membrane with them. When they do that, we say they are enveloped viruses. They have a shell around them. It's our own membrane, all right? That's an enveloped virus. A naked virus lacks that envelope, all right? There is no membrane that's from the host cell, no membrane surrounding it. It's basically just a protein shell with nuclear material inside, all right? But the enveloped virus has the protein shell and then the membrane surrounding it, all right? And inside is its information, all right? So it depends on the circumstances. We can see all of these different types of proteins that make the spikes on these virus, and there's all sorts of variation dependent upon the type of virus as far as the protein profile or the amino acids that set up the stage for this to have its specific shape. Now it's like a lock and key, where this specific spike comes in contact with a receptor on a host cell, and it fits. It's not like these host cells have these receptors saying, "Come on, virus, I'm looking for you." These are receptors for different things, different molecules in our body. But the virus knows that, and it's like, oh, if I have this specific looking piece, I will fit right in there. I can do that. And that's how they are able, well, to kind of sneak inside of these cells, if they will, by making a cell think that it's picking up something that it actually wants, but it doesn't, right? You didn't want this. You didn't want to bring that in, right? Pretty sure you're not looking for HIV to infect your cells, right? Don't think you want that, right? So there are all sorts of crazy morphologies, shapes of virus, and it can be really complex, like that bacteriophage we looked at. It can be pretty simple, like maybe the tobacco mosaic virus right here. It's more of a helical shape with amino acids circling around it, forming this tube, and inside the DNA or RNA, depends on the virus, is packaged up. And we'll get to the nuclear material in a second. Here's rhinovirus, human rhinovirus, and it's an icosahedral virus. Rhinovirus is one of a common cold-causing virus. There are a plethora of cold viruses, hundreds of different types of cold viruses. Rhinovirus is one type. Coronavirus is another, right? Common cold-causing virus, right? Or maybe pandemic proportions, all right? It depends. Variola over here, these guys more kind of, it says complex, it almost looks sort of bullet shaped. That's the rhabdovirus for rabies. This one's not. So variola, we will talk about in a bit. There are a handful of different potentials with the variolas as far as who they might be. But stuff like chickenpox fits into that category. So we're shifting back to some bacteriophage. And as we talk about this, although we're looking at bacteria and what's happening, there are definitely some similarities when we think about virus infecting animal cells as well, all right, as far as what's happening, all right? But again, we're looking at bacteria. So this cycle is called the lytic cycle. And I told you there's two ways that virus might get in there, all right? So this first one is the just straight up, I'm going to infect you and kill you, is basically what's up, all right? The next one is a little bit of persistence. So the first thing that happens, you've got your bacterium, and we've got our circular bacterial chromosome inside. We're not showing you anything else, just bacterial DNA. The virus comes in contact with that host cell. It's the proper cell. We dock, and now this is the attachment, the docking, right? As we land on this cell, everything's all fine and good. If it is true, it is all fine and good, we move into this next bit. And those tail pins on that sheath poke into it, and that's the penetration phase. As it does that and basically drills a hole through the cell wall of that bacterium, it then allows the nuclear material, in this case viral DNA, gets injected into the host cell, all right? The DNA gets in here. As we do this, once that cell gets infected, typically the bacterial chromosome starts to get degraded. It starts to kind of get shut off. We don't want that bacteria going on being a bacterium anymore. We want it to be a virus factory. So we don't want you trying to go through a process of replicating or anything like that. Just make virus, all right? So as we move into the biosynthesis, you don't see any bacterial chromosome anymore. We've pretty much just tried to wipe it out. So now we've just got a factory that's going to make more of the virus, all right? So biosynthesis is the next step. The phage DNA replicates. We produce the phage proteins using the bacteria's enzymes, using its machinery to see this structure, the nucleic acid, and say, oh, let's copy it. And as we copy it, we will also make some messenger RNAs that say, build the capsid, the protein shell. It builds all of the proteins. They self-assemble. You get all of these pieces and they come together. That's brilliant as well. As they self-assemble, we then mature, we package up the nuclear material into the phage head, and now you've got a baby virus, if you will. At that point, once these have all matured, the cell lyses. It explodes, if you will, releasing the virus to basically go on and infect new cells, right? And as I said, single virus in and you get a handful of virus out, right? In this circumstance, right? So it is really a pretty rapid process from the time they dock to the time it takes to mature and rupture out of there, all right? And we can witness some of this stuff growing them up in a laboratory situation. You can see the effects like the following day in some of these plates. And I think I got an image that will show you some of that. Okay, so this next frame is the other idea, all right? Temperate phages have, well, they've got the lytic cycle, the one I just showed, but they also have the lysogenic cycle. And this is what I was describing, where they basically kind of lay in wait, right? We're going to hold off. We're going to replicate in these bacterial cells and ramp up the population of bacteria that harbor the virus, all right? So instead of this, where we just make a bunch of virus, handful of virus, to infect a handful of other bacteria, we're going to basically keep these populations just growing, right? So we get lots of virus potentials because of all of the bacteria as they've replicated, all right? So similarly, that bacteriophage, just like here, it attaches, it penetrates, right? It's doing all of that, all right? But instead of going through the next step, it incorporates into the chromosome. So the blue is the viral, getting into the pink, the bacterial, all right? And as this gets incorporated in, we now say it's a prophage, this virus inside of the bacterial chromosome. As it divides, the prophage DNA gets copied, and now we've got 2 cells, or 4 cells, or over and over and over. It's exponential growth with the bacteria, so you can have massive amounts that all harbor the same instructions. Build me some virus, all right? Now, after we've gone through numerous rounds of division, there's something that triggers this mechanism to say we ought to move on, right? Whatever it might be. It could be a stressful circumstance. It could be a reduction in nutrients that might mean these bacteria are starting to slow in their replication. And if that starts to slow, maybe that's enough of a stimulus to say we ought to move on, right? The prophage, the viral DNA gets excised, so it gets removed out of that chromosome, which is what needs to happen to basically make it go back to this bit right here, all right? To the starting bit where we're going to just synthesize and explode, all right? But again, we're talking millions or billions of bacterial cells that are harboring this stuff instead of 1, right? So it excises it, and then we move into the lytic cycle. So we go through biosynthesis, we create the capsids, we replicate the DNA or the nuclear material, we assemble them up, and then those bacterial cells lyse, releasing out more phage, and the cycle continues, all right? So these are more of a situation where they get in and they kind of go dormant, right? They lay in wait. There are viruses that plague people that we see also some latency. It goes dormant and flares back up potentially, right? It's not the exact same idea here, but it's a similar idea where it is persisting, right? Some, cold sores, herpes virus is an example of something that is, well, it's the gift that keeps giving, right? You never get rid of those. And the potential for it to come back is always there, right? So it's not the same, but it kind of makes me think that as it's laying in wait. So one way or another, they get stimulated to release the virus, and in turn, like the previous slide, that bacteria is going to die as it lyses, releasing more virus to go on. So different as far as how they impact these cells. There are ideas of, and we've talked a little bit about this stuff, and I've definitely seen some stuff in the packback threads talking about some virus and phage and things like that. There are some ideas of using bacteriophage to treat superpathogens, superbugs, things that are antimicrobial resistant. We don't have any drugs to treat these things. How are you going to kill them? Maybe a virus is what we could utilize, right? And as I said a minute ago, the virus, the bacteriophage, is highly specific, targeting a certain type of bacterium, typically only that type of bacterium. So if you could find a phage that truly focuses in on a pathogen, and that pathogen is plaguing you by applying some of this bacteriophage to the individual, whether it be ingesting, or putting it into a wound, whatever it might be, depends on where the infection's at. If you could get the bacteriophage delivered to that area, they will target the bacteria and they will destroy them, all right? With that said, you want them to be these guys, right? You want it to be the lytic type of virus. You don't want it to go in there and just wait, right? Because then you're not really targeting that infection. You're just letting it persist within there. But if you can get a lytic virus and it can target that bacterium, it's going to destroy them, all right? And potentially cure you of that infection, right? In the United States, it is used only under special circumstances and has been used very, very rarely, but it has been used, all right? Other countries, it's a different story, all right? And potentially it's the standard of care and not antibiotics, all right? And that makes you wonder, what are we doing, right? Maybe we should focus some more research into this, all right? And we definitely, well, we are thinking about some of these things. And I've mentioned C-phages in here before. That is not exactly the idea with C-phages, to find some virus that's going to cure these pathogens, but it kind of is. That's on the back burner, right? As we are evaluating all sorts of different possibilities, we are looking for bacteriophage that target specific types of bacteria, and some of those bacteria could be pathogens, right? But it's not, the end goal isn't to go out and just try to find something that's going to cure some illness, all right? It's to get a broad overview of the diversity of bacteriophage, right? There, it is mind-boggling to think about all of those possibilities. It's the tip of the iceberg as far as what we know, right, about virus, and definitely exciting. There are other concerns, and these are things that, initially that I sort of thought about a little bit, with stuff like drug resistance And as we bombard our bodies with antibiotics to try to treat off some of these pathogens, issues happen, whatever it might be, misuse, overuse, you don't take all your antibiotics, whatever it is. There are numerous things that set the stage for some of these bacteria to mutate and to persist and to cause problems, right? So if we can see antibiotic resistance, it makes me a little worried that maybe we would see these bacteria developing resistance to the bacteriophage, right? That was part of my thoughts. But then we back it up for a second and think about these things in a little more detail. And bacteriophage and bacteria have been around for a super long time. I'd like to say for forever, all right? And in turn, virus mutates. We know that in this stinking pandemic, virus mutates, and it depends on the type of nuclear material lending to greater or lesser amounts of mutation, but it mutates. A bacteria might mutate to potentially lend to some resistance, but that bacteriophage also might mutate to in turn open up new potentials to continue to infect. So resistance, although it might be a potential concern, is not as big of a concern as something like an antibiotic, all right? I still feel like there might be some concerns, all right? But not as much, all right? And I think that's pretty impressive, right? Just to think about those possibilities, all right? So here, this is a little bit different, all right? So transduction is a mechanism where we may get some genetic exchange, right? There are really, when we think about bacteria getting some sort of diversity, some sort of new stuff going on, there's three ways that bacteria can pick things up from outside, if you will, all right? Bacteria can conjugate, where one bacterium has a plasmid extra chromosomal material, and it comes in contact, close contact with a neighbor, and they conjugate, and they will form a pilus contacting the opposite cell and injecting some of this material, the plasmid, into that cell, all right? I read something just the other day talking about some bacteria sharing through conjugation, B12. Different bacteria needing these vitamins, but one's able to pull it in and they give it to others, all right? That's pretty cool stuff, all right? So bacteria can get extra material through conjugation. They can get extra material through a process called transformation, where they're just like little recyclers, little scavengers going around picking stuff up, all right? And if they find something of value, they will pull it in. Nucleic acid, that's valuable resources. We could use that to replicate, right? We need those ingredients. So if it comes across a chunk of DNA, it might like to pull that in. Why waste my time building all of these nucleotides? I just found a pile of them, right? I'm going to pull that in, right? Well, maybe that pile of nucleotides actually has a chunk that's an intact gene. That piece could merge with the bacterial chromosome, and now it's got something that it never had before, right? Transformation, we say, is the uptake of naked DNA. The third process is up here, all right? Transduction is when a bacteriophage injects bacteria chromosome into a different bacterium, all right? And it happens like this. So same idea, that virus infects, it gets in there, all right? And we're going to incorporate, so we're thinking lysogenic that we just described. As we get in there, now we're part of that chromosome. As we get excised, as we get chopped out to kick into the viral replication phase, potentially we get chopped out with a little bit of bacterial chromosome as well. So now we're taking more with us, all right? And this stuff, can get packaged up, and it can incorporate into this chromosome new bacteria, all right? And it's hard to see, but it's got viral and it's got bacterial chromosome, and now it incorporates, and this bacterium is now modified. It's different. It's a recombinant. It's got other bacterial information incorporated into it. And part of the viral stuff is probably absent, or maybe completely absent. We didn't bring any of that, but we brought a different gene in, and we can see all sorts of craziness with that as well. All right. And I'll leave you with that. Farewell. Wash your hands.